Welcome to this session on day number 2 of the forum. We're going to be talking about hilltops to oceans today. We're going to be discussing this relationship between marine and freshwater systems and how most of the pollutants in the ocean actually originate on land, borne by freshwater and the atmosphere to the sea. We're going to have people to explain this to you. We're going to also explore how nuclear techniques can assist the industry and the authorities to reduce the impact of land-based sources of marine pollution. Now on to Richard Bernati.
Good morning. Can I have the first slide? As you've heard from the previous talks, we are extremely good using nuclear techniques to measure minute quantities in high dilution. The question is, how good are we at actually measuring what complex biological systems are doing? And before I go through demonstrating how information heavy a feather-light object like this is, I would like to ask Naga to pass around a parcel and have a look at it, just to have a look at it and guess the weight of its content. And if you're really, really curious, you can throw your business card in, and then my colleagues who know much more than I do will get back to you. Now, the feather that you have just seen is from a flesh-footed shearwater. This is an apex predator, a top predator like humans. And that feather was picked in one of the most pristine areas in the world, Lord Howe Island, which the bird reaches on its yearly migration across the equator in a huge figure of 8 from Japan down to Tasmania. This is Lord Howe Island, and this is where our colleague Jennifer Lavers does field work, and it is field work in the literal sense, and plucks from the chest of usually a shearwater chick a feather. Now this feather is then sent to us at the Australian synchrotron, but before I go on describing what we measure, just look at that feather. You see the center part, the stem, it's called the rachis, and then you have a branch, it's called the barb, and from that barb branch of other branches, the barbules. And that is a very almost fractal geometry, highly complex, the mother of all Velcros, really. Now, how does it grow? You have a bundle, a little heap of undifferentiated cells. You look onto the skin, and in these little nests you will find a couple of cells that are already slightly different. And as this little cumulus grows and grows into the shape of a feather, you see these little red dots symbolizing the small cells growing back to base. And as they're growing back to base, they start rotating, twisting, and fusing. A very complex, coordinated process. All along, then you have another set of cells growing from the base to the apex, to the tip of the cell. Finally, you end up with a skeleton that unfolds like a leaf, and there you have your feather. So it's an extremely interesting process that demonstrates two things. First of all, something that is a genetic program, but at the same time is immediately influenced by the local environment, the next cell. So there is evolution and development. And in the lingo of biologists, that's the evo-devo question. What is genetic? What is development? From the field work to the pristine environment of the Australian synchrotron, as I've already mentioned, where the feather is suspended in a beam of electrons accelerated to relativistic speed of light, hence it's also called a light source, and that reveals something rather interesting. So these are 3 different feathers, and the top row is the distribution of the element bromine. And you can see there are areas where the bromine is high and low. It's overall quite irregular. In some cases the stem, the rachis, is high, but more interesting is the next row of the same feathers, but now concentrating on the element of zinc. And as you can see, I hope you can see that, maybe I'll look here, you see these bands, almost like tree rings. So first we thought that was an interference pattern, an artifact. No, that is indeed the case. Now, how does one arrive, how does nature arrive in what is a simple gray feather at this very regular pattern. Here's just another illustration. Just go back to the animation. You remember the feather grows, these red cells go up, and all along there is a pulse, a daily pulse of zinc in the blood of the animal. And that's called a diurnal variation. How do we know it? Because if we count the stripes, these are around 28 to 30 stripes, which is the time the feather needs to grow. So now we can ask the question not only who done it, but also when done it. So, as you know from dating exercises with tree rings, having structure, repetitive periodicity in biology is a major part of understanding how a system responds. Now, from the synchrotron, and it is the very same material, everything is non-destructive, that feather came to the OPAL, Open Pool Reactor at Lucas Heights near Sydney, to perform neutron activation analysis. The reason is, as I've already mentioned, there is an environmental stress on these small shearwater chicks. Their bellies are full of plastic. And now you can look into the plastic bag, whoever has it. That's roughly the equivalent that is in a single chick that you have in your plastic bag. And upscale to a human, that would be 2.5 kilograms in our belly. That it doesn't always end well is almost self-evident. So there is mortality just from the sheer volume. But we were interested in whether contaminants in plastics would transfer into our feather as a readout at a single individual level. Just to remind you, neutron activation is the bombarding, again, non-destructively of an object, here the feather, and the emission, the gamma emission, allows us to see the profile of elements. We did the same with the plastics. Here, this is the content of a single bird, just sorted by size. And the plastic content or the stomach content roughly has that distribution pattern. It's not important to look into the individual elements, but just look where the red box is that contains titanium. And now go to the next slide. You see the overall distribution. You go down from the stomach content to here on the left. This is a bird that has looked healthy and had no plastic. And indeed, titanium, it's just sort of a lead isotope, is not, or lead element is not present. However, birds that had either plastic or signs of physiological stress would show increase in titanium. Now, titanium is a white plastic, white powder. It's not actually toxic in itself. This is why you have some marshmallows in the plastic bag as well. So we don't exactly know what the source of the titanium is. We assume it's in plastics because that's where it's used. So I summarize, we've used two nuclear techniques, synchrotron radiation, to address this very fundamental biological question, evolution, development, and then neutron activation to relate it to an environmental stressor. That brings together, of course, physicists, engineers, life scientists, and very importantly, a huge number of highly dedicated individuals, citizen scientists, who do collect in a very rigorous manner the plastics around the coast of Australia and also internationally. If you have any questions, you can either send a carrier pigeon, Shearwater, or just send an email. Thank you.
Thank you very much.